Okay, hi everyone. I just want to talk to you guys about meiosis and sexual reproduction. And this is the prelude to talking about Mendelian genetics, which you're going to be doing towards the end of the week with a major homework assignment there. So meiosis is an outgrowth of mitosis, and it's essentially a way that a cell that's going to be involved in a sexually reproducing organism to produce gametes, which are cells that have half the number of chromosomes that a normal cell in a body would have. And we'll get into that a little bit in a second, what that really means. Um, so um, some terms you need to understand to get meiosis, things that we kind of bat around in class a little bit, but should be understood in a more formal way as we go forward into genetics. The idea of the homologous chromosome is the first thing we talk about. This is the idea of that there in any organism that diploid, and we'll talk about what that means in a second. There are two nearly identical chromosomes that make up a matched pair in a diploid organism. That means that for every chromosome that organism has, there's a second one that is similar. Not identical necessarily, but very, very similar. It has the same genes, same array of genes, more or less. And usually you get one homologous chromosome from each parent in your when you're generated during sexual reproduction. So you'll have one homologous chromosome from one your mother and one from your father for each set of the chromosomes. For humans, for example, we have 23 sets of homologous chromosomes, or 23 pairs. And so we have one of each chromosome one from your mother, one from your father, and then you have one chromosome 2 from your mother and one from your father, and you have two chromosome 3s, two chromosome 4s, two chromosome 5s, et cetera, all the way up to the sex chromosomes. So that's the homologous chromosome. Sex chromosomes are those chromosomes involved in determination of sex in an organism. And this is in humans, this is the X and the Y chromosome. They're considered unique chromosomes because they're determined in sex, used in sex determination in mammals. X chromosome is an important, very important chromosome, has lots of genes that are not involved in sex determination, and both males and females need an X chromosome to survive. The Y chromosome, on the other hand, is a very, very minimal chromosome, has only a few genes on it, and one of those genes is a gene that's really required to switch on the male pattern for development. So that chromosome is Y chromosome is only found in males. Autosomes are all those chromosomes that are not sex chromosomes. So humans, we have 22 pairs of autosomes, right? So we have 23 total pairs of chromosomes, 22 autosomes, and one sex chromosome pair. A diploid genome is a genome that contains pairs of homologous chromosomes. So, right, in other words, two of each type of chromosome, which I just talked about. So then the haploid genome is a genome that contains only one of each type of chromosome. And during meiosis, what we're going to do is take a cell with a diploid genome content, and divide it until it has a haploid genome content. And then during sexual reproduction, you take two haploid cells, an egg and a sperm, and fuse them together to make a new diploid genome in the new cell, the zygote. All right, so that's sexual reproduction, and that's why we need meiosis to do that process of going from diploid to haploid genomes. All right, so that's what sexual reproduction is. Reproduction requires a fusion of two cells containing haploid genome content, sperm and the egg, and they form a new cell containing a diploid genome content. The gamete are those cells that are involved in carrying out this fusion of haploid genome contents, right? They're generated during meiosis, and they contain a haploid genome content, sperm and the egg. Fertilization then is the fusion of two gametes, sperm and egg, to form a new diploid cell, which is a zygote. And the zygote is a diploid cell that results from fertilization. All right, so these are the important terms for meiosis and sexual reproduction. All right, so here's our life cycle as humans. Right, up here is the haploid cell stage, and down here's the diploid cell stage. Right, so most of our time, and most of our cells in humans are diploid, and almost all the cells that make up the human body are diploid cells, except in those cells, a few that are haploid as a result of meiosis, that are going to be in the ovaries and the testes. So the testes and the ovaries produce haploid cells by meiosis, this process here. And in males in the testes, that process of meiosis during gametogenesis is going to produce a haploid cell, sperm cell, right? And in females, that process of meiosis is going to produce a haploid ovum or egg cell, right? The genome content is conventionally indicated as N, N being the number of haploid chromosomes that a cell will have. So an ovum in humans will have an N of 23, right? Right. So sperm cell also have an N of 23, right? Then when you fertilize these during sexual reproduction, you get an egg and a sperm, they fuse, bring those two haploid genome contents together. During fertilization, you make a new zygote. Now it has a diploid genome, and that's indicated as 2N, right? 2 of each chromosome, 23 chromosomes, so it's 46 total chromosomes. And this will go through mitosis and development, through gene regulation, so on and so forth, like we talked about, and you end up with an adult organism in the end, and then you continue the cycle in the gametes of that adult organism. Okay, so this is not the only type of lifestyle there is out there. This is what animals do. That's what I just showed you in a very schematized way. There's other ways of doing this. We talk about plants in the future. Plants actually have something called alternation of generations, where there's actually a form of the plant that's actually haploid, and it's a multicellular plant, actually. And then there's a diploid form of the plant, all right? So there's a diploid form. That's most plants look like they're that you see around are diploid, and then they'll go through meiosis, produce spores, and those will germinate to produce a multicellular organism that's haploid. That's called a gametophyte. And then that will go through mitosis to produce gametes, and they'll fuse two gametes together, fertilization, making now a diploid organism again, and that will go through mitosis to produce a new diploid multicellular organism. It's called sporophyte. Right, and then fungi and some algae do some other things. For example, the main multicellular organism is haploid, right? And then they'll produce gametes by mitosis, fuse them together, fertilization. They'll make a zygote. The chromosomes will be rearranged and so on and so forth. And then they'll go through meiosis and germinate into a new multicellular organism. So the main organism is actually haploid. The diploid form is just one cell, the zygote. Okay, so what is meiosis then? It's a process that produces haploid daughter cells, those are usually gametes, from diploid starting cells, right? Generally, one diploid cell generates 4 haploid daughter cells during meiosis, right? So how does this work? So at some point during the cell cycle, right, you're going to go through S phase. And so you're going to have one chromosome, one of each type of chromosome. One, say the blue is paternal and the red is maternal. So from the parent of this organism that has this cell, they have one chromosome that's from the father, one from the mother. Now it's going to be replicated during S phase. So you have 2 of these, right? 2 copies, 2 sister chromatids for each of these, right? So now there's 4 strands of DNA for each set of chromosomes, right? And those are called homologous pairs. So homologous pairs of replicated chromosomes. They're sister chromatids. But these 2 chromosomes, the paternal and maternal chromosome, are homologous to each other, as we talked about before, right? So this is a diploid cell with replicated chromosomes. Now, during meiosis 1, these homologous chromosome sets are going to separate from each other, all right? So this homologous set, the paternal homologous set, and the maternal homologous set are going to separate during meiosis 1. And you're going to get a cell that's going to have 2 chromosomes in it, but they're going to be identical copies of the chromosome that's in this cell, and same for here. So the paternal chromosomes over here in this cell, and maternal on this side, right? That's meiosis 1. And then we're going to go through meiosis 2. These chromosomes are separated and to produce cells that now have only 1 copy of DNA of the chromosome in each cell, right? So 4 cells, each of which has an N of 1, right? And they started with a cell that had a 2N of 2, right? Or an N of 1. Right, so we go 2 chromosomes, two chromosomes, a pair of homologous chromosomes. Each are replicated during S phase of the cell cycle to make 2 versions, and then they're in meiosis 1. They condense. You have sister chromatids, as we saw in mitosis. Meiosis 1, these sister chromatid pairs separate from each other, right? As sister chromatid pairs, and then during meiosis 2, those sister chromatid pairs separate the 2 strands of DNA from each chromosome into separate haploid cells, right? All right. So how does this actually happen in reality? Basically, very much like we saw in mitosis. We have a cell here. It's gone through S phase. We have 2 copies of each chromosome in the nucleus, right? Then we go into prophase. The chromosomes condense. They form their characteristic shape, right? The homologous chromosomes now pair up. So we have replicated chromosomes for each pair of the homologous chromosome, right? So here we have the maternal chromosome in red, paternal chromosome in blue, right? And then we have another set of chromosomes in this image, right? So this is a organism that has an N of 2, right? And we have a homologous sister chromatid pair here and a sister chromatid pair, and these are homologous paternal and maternal, right? Now what they're seeing here in prophase 1 is that something happens here where chromosomes exchange DNA material, right? They exchange segments. So when these chromosomes align, they line up like this, and it's called a tetrad. These chromosomes actually physically line up. The inside chromosomes in the homologous chromosome sets actually line up and next to each other physically, so they're close to each other. And then the DNA is broken between these 2 chromosomes at one location, and the DNA is swapped and then resealed, so that now you have some of the father chromosome on the mother chromosome on this side, and some of the mother chromosome on the father chromosome on this side, right? You do that for all chromosome sets. So each tetrad will have recombination happening in there, crossing over, they call that also. That happens during prophase 1. Then in metaphase 1, the chromosomes will line up, the spindle's formed, right? Already formed actually during prophase, and the chromosomes will now line up in the metaphase plate, and then the sister chromatid pairs will separate during anaphase 1. Now look, see the maternal chromosome homologous pair has a little bit of paternal DNA on it. Paternal DNA chromosome homologous set has a little bit of maternal chromosome on it, and vice versa over here, right? So we've had recombination happen or crossing over, and the chromosomes are separating during anaphase 1 as sister chromatid pairs. Okay, then you go through telophase 1 and form a new nucleus, maybe the cells separate during cytokinesis, and then the cell is going to reorient and actually go through prophase 2, form a new spindle, right, each cell, and then the chromosomes are attached to the spindle, and then they line up metaphase plate as sister chromatid pairs, right, and then these release from each other, and these sister chromatid pairs separate into separate chromosomes, and you get now 1, 2, 3, 4 cells, each of which has 1 copy of each chromosome. So N of 1 for each of these, right? Okay, so that's a simple process. If you want to watch this actually, how it happens, animated, use this site, and actual cells doing this stuff, you can actually look at these 2 links here. You may need to copy and paste these links into your browser to see them. You'll see me working out PowerPoint right now. Okay, so what's basically happening here? So here's we're going to compare mitosis and meiosis are very similar, right? Start with parental cell. It has a chromosome content of 2 ends, diploid of 4, right? So it's 2 pairs of homologous chromosomes. During S phase, the DNA is going to be replicated. So in mitosis, each chromosome has two pairs of DNA, sister chromatid pairs, right? And they're going to line up during metaphase, and then get separated during anaphase and the telophase, and then cells separate during cytokinesis. You end up with 2 cells, each of which has an identical copy of DNA of 2N, right? Because this is replicated, but during S phase to make 2 copies of each chromosome, right? So now cells actually essentially 4N in a sense at that point, and then it gets divided into 2N, just like we saw in mitosis. Nothing changed. In meiosis, the cell is going to replicate its DNA during S phase, and then during prophase 1, the cells are going to form tetrads, right? And then cross over along the chromosome sets in the middle of the tetrad, right? And then metaphase 1, they're going to line up the metaphase plate. Homologous chromosome pairs, sister chromatid pairs, are going to separate, right, during anaphase 1, telophase 1, and then during meiosis 2, those homologous chromosome pairs are going to separate to separate cells, right? We get 4 cells, each of which has 1 N chromosome content. Okay, so this just summarizes what happens during each of those phases and what doesn't happen in some phases and why it happens, all right? All right, now, what's the point of this idea, of this process? This supports a way of reproducing, which is called sexual reproduction, which is you have 2 separate organisms contributing their genetic material to a new organism. And that results in all sorts of genetic variation. There's all sorts of advantages for why cells do that. We'll talk more about that when we do evolution. But so we can talk about some of the sources of genetic variations, results of sexual reproduction and meiosis right now, right? So for example, independent assortment of chromosomes results in lots of genetic variation. If you have paternal and maternal chromosome sets, and during, even if a cell only has an N of 2 here, which is what this one does, right? During meiosis 1, you get a cell that might have this combination of chromosomes, just paternal on this side and maternal on this side after it goes through meiosis 1. Then you go through meiosis 2 and you get haploid cells which have just paternal chromosomes or just maternal chromosomes, right? Or you could have the possibility too for a simple organism like this, that where the chromosomes line up the other way. So, right, so now you have paternal and maternal lined up on one side and maternal and paternal lined up on the other side. So when you go through metaphase 2, you end up with these variations. Now the mother's DNA, the original mother's DNA content, right, is going to be in this cell for this chromosome and in this cell for this chromosome, and the paternal is going to be vice versa, right? So now when you go through gametes, now you have a mix of maternal and paternal chromosomes here, right? Okay, so for a cell that an organism has 2 to 23 different chromosome sets, right? That actually means there's 2 to 23 different possible combinations of chromosomes. It's 8 million different combinations. That's a lot, right? This does not include crossing over. Crossing over also happens here too, right? That adds additional variation right here. So here's crossing over. Crossing over adds additional variation. You don't know where the crossing over is going to happen in here. It can happen anywhere along the length of this chromosome. It can happen close to the centromere, or it can happen towards the tip. You don't know, right? But it's going to happen. And so all these chromosomes are going to have new DNA combinations as a result. Some of the DNA in the maternal and paternal DNA can be combined in new combinations here, right? So cell gametes are going to have lots of combinations of different possibilities. Then on top of that, you have this independent assortment, which adds additional complications and variation, right? And then also random fertilization means any egg can be fertilized by any sperm. So it's 2 to the 23 possibilities of different diploid of different chromosomes in one gamete, 2 to 23 in the other gamete. That's multiplied together. That's 70 trillion different possible combinations of chromosomes. And does not even account for crossing over, right? So this is a lot. There's not even been close to 70 trillion humans on the planet so far. So this is way more possibilities than we even have probably mammals that have ever lived. So I don't know. Yeah, it's a good question, actually, how many mammals have ever lived. But this is a lot of variation, right? So even if you have a brother or sister, say you're female and you have a sister, you have a 1 in 70 trillion chance of being identical to that sister. And that doesn't even count for crossing over, right? So there's actually a lot of variation here, right? Then you have random mutations also, right? You have point mutations and various other types of deletions, mutations we talked about also that happen during DNA replication during S phase, right? Those are introduced in the gametes also, right? So that adds additional variation. So you get lots of variation that evolution can work with in any combination of gametes. All right, so that finishes that part, and hopefully they'll get you through what you need to do this week, and then we will continue talking about this for my next video when we talk about how this applies to Mendelian genetics.